Hello, I am Komudi Singh from FDA's Office of Therapeutics Products, and today I'm here to introduce you to a recently published draft guidance called Safety Assessment of Genome Editing in Human Gene Therapy Products using Next-Generation Sequencing. This draft guidance provides recommendations for how developers of human gene therapy products incorporating human genome editing can use next-generation sequencing technologies or NGS technologies in nonclinical studies to assess the safety of these products. My goal is to cover some key points from the draft guidance that include summarizing the scope of the guidance and introducing the safety risk associated with off-target editing and genome editing impact on genomic integrity by providing a high-level overview of the content of the guidance. I will not cover everything, so please refer to the document for more details. My other goal is to encourage you to provide us with your feedback on this draft guidance. You may provide your comments or feedback in the docket on the website where the draft guidance was published. FDA published a guidance in January of 2024 that provided broad CMC, non-clinical, and clinical recommendations for human genome editing products. The newly published draft guidance adds to the non-clinical considerations discussed in the previous guidance and elaborates on recommendations pertaining to the use of NGS for safety assessment of human genome editing products. The field of genome editing has significantly expanded to include diverse editors that can edit DNA or the epigenome or cleave RNA for gene expression downregulation. Since safety assessment of these diverse editors also use NGS technology, the recommendations made in this draft guidance are applicable to DNA editors as well as epigenetic editors and RNA editors. One of the risks associated with genome editing products is the risk of editing at a genomic site not intended for editing. If the off-target editing occurs within regulatory elements, then it can potentially disrupt regulatory function, which may be deleterious to cell function. Similarly, editing within the gene coding sequence can potentially disrupt gene expression, which may be deleterious to cell function. Hence, this risk should be appropriately assessed. An unintended editing outcome at the on-target edit site can also result in a failure of gene correction or disrupt gene function and should be assessed. Additionally, an erroneous repair at double-strand break sites may result in chromosomal rearrangement events that can impact genomic integrity of the cells. Hence, the risk associated with off-target editing and chromosomal rearrangement events should be assessed using sensitive and quantitative NGS-based methods. Before I dive into the content of the draft guidance, I will briefly cover the key topics presented in the document. The draft guidance provides recommendations for use of NGS in non-clinical studies usually implemented to assess safety of genome editing products. Specifically, I will summarize recommendations pertaining to NGS use, recommendations pertaining to on-target editing, nomination and confirmation of off-target edit sites, sample selection, reporting of unintended editing outcomes with appropriate risk assessment, assessment of impact of human genetic variation on off-target editing risk, assessment of chromosomal rearrangement events, and recommendations on submission of study reports and data to the agency. With rapid advancement in the field of sequencing and bioinformatic methods, there are many NGS-based methods available for assessing the genome, epigenome, or the transcriptome. The recommendations made in the draft guidance provides considerations for selecting an appropriate method that meets the goals of the analysis. For example, sequencing needs for a genome-wide analysis would be distinct from that of a targeted sequencing study. Use of adequate input material measured as cell number or genomic material would facilitate adequate sampling of cell genomes. Use of appropriate sequencing depth would allow detection of low-frequency changes occurring in a subset of cell population. Additionally, sponsors should determine and apply appropriate sequencing-related quality control metrics to determine sequencing data quality, document tools used, document analysis steps, and record results. All this information should be submitted to the FDA. Next, the draft guidance covers considerations for on-target edit site and off-target edit site analysis. The editor mechanism of action should inform selection of appropriate NGS methods for these studies. When modifying NGS methods or published off-target assays, sponsors should document the modification, report the changes, and provide data to support that the proposed change did not impact the assay sensitivity or introduce bias. Consistent with published guidance on human gene therapy products incorporating human genome editing, off-target edit site nomination using multiple methods is recommended, that I will further expand on the next slide. Later in the presentation, I will further discuss assessing the impact of human genetic variation on off-target editing risk and impact of genome editing on chromosomal integrity. As I stated before, consistent with published guidance on human gene therapy products incorporating human genome editing, use of multiple methods for off-target edit site nomination is recommended. For some editors, there are established biochemical or cell-based methods for off-target edit site nomination. Specifically, for some editing modalities, a biochemical assay, as shown in the top left cartoon, may be used where the genomic DNA is edited in vitro, followed by sequencing and analysis. For cell-based off-target analysis methods, as shown in the middle left cartoon, cultured cells are edited, followed by genomic DNA extraction, sequencing, and analysis. However, with rapid expansion of genome editing tools, there may not be established methods for off-target editing analysis for every editor. The sponsor may use other generally applicable methods for off-target analysis. Specifically, the sponsor may use computational or in silico methods to identify sites in the human reference genome that are similar to the proposed guide RNA or the DNA sequence being targeted for editing. Additionally, appropriate NGS-based methods selected based on the editor mechanism of action are recommended. Sponsors should provide additional data and justification to support the adequacy of NGS methods used. For more information on considerations for off-target edit site analysis, please refer to the draft guidance. The draft guidance also describes the list of information that should be submitted to FDA when reporting the nominated and confirmed off-target edit sites or identified chromosomal rearrangement events. When reporting an off-target edit site, the associated read count and editing frequencies as applicable should also be reported. Additional information includes genomic coordinate information, alignment information between the edit site and the guide RNA or the target DNA, and report any differences as mismatches or bulges. Annotation information should be used in conjunction with published data if available to assess the functional consequence of the edit. Additionally, gene expression information from cells or tissues may be used to discuss potential impact of off-target editing on cell function and for providing risk assessment. Since the off-target editing risk is impacted by the cell's chromatin landscape, which would impact accessibility of editor to the genome, use of appropriate samples for assessing ex vivo and in vivo products is necessary. For in vivo genome editing products, biodistribution data should be used to determine the need to assess additional cell types where the product was detected. Use of multiple replicates of human cells is recommended. Additionally, human cells harboring a target sequence would facilitate evaluation of editing activity at the target site, as well as assessment of off-target editing risk. The editing parameters and editing delivery methods should be appropriately selected to be representative of the methods used in the final drug product. I encourage you to read the draft guidance for more information on sample selection considerations for ex vivo and in vivo genome editing products. An individual genome is known to harbor millions of nucleotide variations when compared to the reference human genome. It is possible that a subset of these variations can impact the alignment between a genomic site harboring the variant and guide RNA by making the site more amenable for editing. For example, as shown in the cartoon, presence of a variation may decrease mismatches between the variant harboring site and the guide RNA. The off-target editing risk associated with these variants should be appropriately assessed. For this, in the draft guidance, recommendations on database selection and variant selection criteria, analysis approach which usually includes computational methods to assess the impact of variant on off-target editing risk, and reporting of findings of these studies are covered. Sponsors are recommended to provide justification to support their strategy for database selection and for any variant filter step based on variant allele frequency or other criteria applied. For genome editing products utilizing double-strand breaks, erroneous repair process may result in chromosomal rearrangement that may be detrimental to genomic integrity of the cells and should be assessed. Use of quantitative NGS methods is one way to assess genomic rearrangement events at these sites. This analysis should be performed at on-target site and all confirmed off-target edit sites. Reporting of these events should include genomic coordinate information, associated read count or frequency information, and annotation information along with risk assessment. Sponsors are encouraged to discuss their study plan and strategies with the FDA using meeting options such as Interact or Pre-IND prior to IND submission. We recommend that safety studies using NGS be completed and study report with results be submitted in the IND application. Please note that additional analysis may be needed if there are significant manufacturing changes. For example, when a manufacturing change involves a change in editing parameters resulting in significantly different intended editing outcomes, additional studies to assess and report off-target editing outcomes may be necessary. Additional data, such as raw sequencing data and analysis scripts, may be requested during clinical studies or BLA application. In closing, I would like to thank you for your time and interest in this topic and encourage you to read the draft guidance and share your feedback with us that can be provided at the docket number listed on this page. The recommendations in the draft guidance are provided to help stakeholders with planning and implementing appropriately designed non-clinical studies and using appropriate NGS strategies to adequately assess off-target editing risk and the impact of genome editing on genomic integrity. Your input is important, and we hope to receive your feedback. Thank you.